Hello, this is Kelly Eversole from the International Wheat Genome Sequencing Consortium, and I'd like to welcome all of you to our first webinar. We've launched a new webinar series this year, and you're on for the first one. So just to remind everyone, we have almost 3,000 members. Anyone can be a member. And I'd like to thank all of our financial sponsors for making this webinar possible. As part of the IWGSC 2.0 vision, our goal is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And that is part of the reason why we wanted to start a webinar series to begin to advance this vision. Our activities in 2020 include quite a bit of activities related to our vision, including the expansion of the IWGSC Arbor Biosciences collaboration, continued effort on IWGSC RefSec v2.0 and our annotation, and then, of course, as I mentioned, launching this webinar series. So today I am very pleased to introduce Yves de Smet from VIB in Belgium, and he's going to be talking today about understanding abiotic stress signaling in wheat through phosphoproteomics. So I want to thank you for your participation, and at this time I'll turn it over to Yves.
So also welcome from my side to this webinar, where indeed I will be talking a little bit about how we approach abiotic stress signaling in wheat through our phosphoproteomics approaches. But before kind of really going into the scientific details, I want to start with a little side project where there is also a little association with wheat that I've been developing with a friend of mine, where we basically try to capture the biological diversity of fruits and vegetables and crops, and such as grain crops such as wheat, by looking at art, predominantly looking at paintings, and we then try to combine this artistic information kind of provided by my friend, who's an art historian, with the molecular biology underlying some of these processes to try and find explanations to, for example, why wheat used to be basically tall. And if you look at this painting of Bruegel in the mid-16th century, you see that the size of this grain crop is nearly the size of a person. In order to do that for a wide variety of fruits, vegetables, and crops, we would like to ask for everybody's participation in going to musea, going to mansions, castles, and look at the art that is presented there, mainly then paintings, and try to send us pictures or information on these works, so we can assemble an amount of material which then allows us to draw conclusions. In the context of wheat, for example, this is when do we see that wheat becomes more or less knee height. So with this little introduction aside, I would like to move into the actual topic of this webinar, where I want to mainly focus on wheat that is currently under stress. There is a number of biotic stresses that are tackling our wheat crops in the field. I will not really go into these. I will mainly focus on abiotic stresses, and then predominantly temperature stress, cold and heat stress, a little bit of mineral stress, and some drought stress is also what we are looking into. And it's becoming clear on why this is important if we are looking at this graph, where we see that there is a dramatic yield loss in wheat, but also in a number of other crops upon abiotic stress. So we really need to understand and tackle this problem. If we specifically zoom into the impact that high temperature has on wheat yields, there have been analysis and predictions that actually say that per degree increase, we are expecting a 6% decrease in wheat yields. So if the climate continues to change as it is changing now, this will have dramatic impacts on wheat yields and overall food security. So we really need to try and understand how plants are dealing with this and how we can essentially prepare plants with these changes in mind. So what we are mainly focusing on is temperature stress. As I said, we are doing this in a number of plant species, including wheat. And in our research, we are trying to look for the really early signaling and perception mechanisms that are associated with these stress conditions. So we're not explicitly looking at the physiological and developmental changes, but we're actually trying to capture the early mechanisms that lead to these responses, hoping to identify a number of very specific factors that are controlling these processes. And I'll come back to that a little bit later. In addition to looking at these early signaling events, we are looking at protein level signaling. So we are ignoring largely, of course, to obtain these proteins, we need to go through the DNA, RNA phases, but we are predominantly focusing on the protein levels. And we are focusing on these proteins as a whole. So we are looking at what is happening with respect to the whole proteome. And we try to kind of capture differences upon different stress conditions. And one added complexity to this is that you can imagine that you have a number of proteins that make up this proteome, this protein pool. And if it would be that simple, it would be reasonably straightforward to try and capture this. Unfortunately, there are a number of post-translational modifications that impact on each and every protein that are either reversible or irreversible, and that include hydroxylation, acetylation, methylation, ubiquitination, and also phosphorylation that control activities, localization, and so forth of these proteins. So all these modifications together in the context of the proteome dramatically and exponentially increase the number of potential proteins that are available. And it is these proteins that were actually controlling, as I said, very specific processes. So what we are interested in the context of signaling at the moment is very specifically one of these modifications, which is phosphorylation. So the activity of kinases and phosphatases that lead to the post-translational modification of predominantly serine, threonine, and tyrosine, a little bit of histidine as well in plants, that will then control the activity, the turnover, the localization, crosstalk, conformational changes, and protein-protein interactions of all of these proteins in plants. So we know that kinases and phosphatases play an important role and that they have numerous targets. But what we are aiming to capture is, of course, very subtle changes in sometimes one particular organ or even one particular cell that will impact on these particular proteins. So in order to do that, we need to really enrich for these phosphorylated proteins. And therefore, we've spent some time in developing and optimizing a workflow that works really well in our hands, where we are starting with the raw plant material, seedlings, flowers, any kind of organ that you can imagine subjected to, for example, a stress condition for a given amount of time. We then extract the proteins and we digested these with specific proteases into small peptides. And these peptides will then be a composition of the proteome, and we have then phosphorylated and non-phosphorylated ones. Since these phosphorylation events are really kind of happening at substoichiometric numbers, we really need to enrich specifically for these phosphopeptides in order to maximize the chances to actually identify these using mass spectrometry, which is then obviously the next step where we do our analysis of the different phosphopeptides by looking at MS2 spectra to gain some sort of idea of the diversity of phosphopeptides that are present in our complex samples. To the extent possible, we also try to normalize the phosphopeptide abundance in the context of protein levels, because you can imagine that a change in protein in the level of a phosphorylated protein can, on the one hand, be due to altered kinase or phosphatase activity, but it can obviously also be due to an overall change in abundance of the protein. So we're trying, to the extent possible, to really capture as much as possible those proteins that are controlled by kinases and phosphatases, which is then giving us insight in conditionally regulated phosphosites. And one of the important prerequisites in order to do this is, of course, a well-annotated genome. So we really need a genome which is then translated into a proteome in order to map our spectra to these proteins. And to illustrate that, the importance of that is that if we look at the recent RefSeq version 1 of the wheat genome, this kind of dramatically increased to 30 to 35% of the identifications in our samples, which seems to correlate with the increase in the number of entries compared to earlier versions of that genome. So this is really a kind of important starting point, that we need a good genome in order to identify and map our phosphopeptides. And once we have all these phosphopeptides, we developed together with colleagues within the VIB this plant PTM viewer, where we not only incorporate our phosphorylation events, but we also have a number of other modifications that are embedded in a number of different species, where you can go and search for your favorite protein and see if you can find some phosphorylation, ubiquitination, and so on in a particular condition. So this is freely available to everybody who is interested in looking at PTMs at proteins. So the way we then approach this in a more specific kind of questions that we're trying to answer is that we look for relevant candidates, and candidates in this context means phosphopeptides that are, of course, belonging to a particular protein. And the data that we get out of that is essentially twofold. So on the one hand, if we look or if we observe the differential phosphorylation of a protein, we assume that this protein likely plays an important role in the process that we are studying. So we use this as a proxy to then try and validate the role of this particular protein in wheat by looking at loss of function lines, for example, and then see if it has a particular phenotype in the context of the process that we are studying. In addition, we also have a second layer of information, because we very specifically know which amino acid was phosphorylated upon a particular trigger. So we can also start to interfere with this amino acid, for example, by mutating a particular serine into an alanine, so it can no longer be phosphorylated, and then we can try to assess the functionality of this particular protein in the context of our process of interest. Of course, wheat is a very important crop, and I think the tools that have been developed, including the genome, including mutant populations, and so on and so forth, in recent years, now really allow us to do functional and rather fast functional analysis in this crop. However, this is still not at the speed of analysis as, for example, in the model plant, the dicot model plant Arabidopsis, where we can then exploit all the information and all the tools that are available in this model plant to really try and gain a faster understanding of the mode of action of our protein of interest that we had identified in wheat. One major advantage, in contrast to approaches in the past where one identified a protein in Arabidopsis and then translated or try to translate this knowledge to relevant crops, is that we are now with our approach are really immediately identifying the relevant candidate in wheat, which we can then very easily implement in functional studies, but also in breeding approaches and so on. So I will now try to give a couple of examples that really look into this methodology in a little bit more detail and kind of give you an overview of what we can actually do with proteomics and phosphoproteomics based approaches. So one example is where we look at a difference in protein level and see if we can correlate this with a particular feature of the wheat plant or the wheat seedling. And for this first analysis, we focused on these three species, one tetraploid and two hexaploid wheat varieties, which at the seedling stage look rather similar, but as an adult plant obviously are different. So the tetraploid and the Capelli one and the hexaploid Pavon 76, they kind of grew up to rather tall plants. The USU Apogee is a variety that was developed to stay rather small and have a fast cycle. So the idea was here to see if we can, at the seedling stage, already identify proteins that are altered in their abundance and that might have predictive power towards how the adult plant will actually look like. So if we look at the proteome for these three species, we see that there is a large overlap of the proteins that we can identify in our analysis, but there are also a number of differences. There are a number of proteins that we only see in either one of these varieties. On the other hand, there are overlapping proteins that we identify either in the tall versus the small one. So this kind of indicates that indeed we might be able, based on a very early capturing of the proteome, to identify proteins that can then predict the future appearance and behavior of this plant. So one example, a couple of examples here in this diagram where these proteins are visualized in this heat map, where we have blue for low abundance and yellow for high abundance. We see indeed that there are obvious groups of proteins that are clearly different between the tetraploid and the hexaploid varieties in this group 2 and group 5. But we also see that there are a number of proteins that are different between the two tall varieties and the small one, both up and down regulated, which is kind of suggesting that indeed we might be able to identify predictor proteins for the growth behavior of these plants. One other example that came out of this data set, and there I focus on one protein that was associated with salt stress, gave us an indication that maybe these two hexaploid varieties might have differential sensitivity to salt stress. And indeed, when we tested this, we could show that one of the varieties, the Pavon 76, is much more sensitive to a salt treatment than the other one. And this basically came out of a differential level of a particular protein that we observed already at the seedling stage. Indeed, further supporting that, we can identify predictor proteins for abiotic stress and growth potential already very early on in these plants. A second example, and this is then much more associated with the main research that is going on in my lab, is the early temperature responsive wheat phosphoproteome that we are trying to capture. So if we look at the impact of an increase in temperature on these two wheat varieties, we see that if we have a rather mild increase in temperature, there is this growth promoting effect, which is comparable with what we see in, for example, Arabidopsis, where the hypocotyl starts to elongate, where the petioles start to elongate, and the leaves start to angle upwards to increase the cooling capacity. Also here we see that these plants, when they are exposed, or these seedlings, when they are exposed to a higher temperature, are growing somewhat faster and are developing somewhat faster. So their leaf development goes faster. At a given temperature, this becomes too high, and we then observe that we see, as for example Chinese spring at 36 degrees, that this temperature becomes really limiting and that we hardly get any growth. So there is this clear optimal growth temperature for these varieties, and this tends to differ amongst different varieties, and I'll come back to that a little bit later as well. Now the idea was to see if we can actually capture the regulatory proteins that are underlying this growth behavior. And as I said in the beginning, we are really interested in early signaling and perception mechanisms. And the reason why we are interested in that is because the assumption that we have is that if we look very early in the signaling pathway, we are able to capture trigger specific proteins. If you have an impact on growth, the general responses, transcriptional changes preceding that, and the transcription factors regulating that are very often recruited by multiple pathways. So there are multiple triggers that feed on this core machinery that regulates growth, but we are really trying to capture the components that are very specifically regulated by temperature. So the quicker we can capture these changes, the more likely we are to find very specific proteins, phosphoproteins, signaling cascades in this environmental stress response. And this really implies looking within the hour, within minutes after exposing plants to this trigger. And for example, here what we did is we exposed seedlings and adult plants to an increase in temperature to 34 degrees. And we already observed that heat shock proteins are very rapidly transcriptionally upregulated. So here we already see very high fold changes after 30 minutes, but we also observed this much earlier than these 30 minutes, which is really suggesting that signaling events associated with this response, this transcriptional response, are preceding this trigger. So we really try to look for changes within the hour. And one experiment that we have done, comparing wheat leaves and ears and spikelets, after exposing them to 1 hour of high temperature, really captures a number of proteins that are very specific for this response. And in order to explain this rather complex result in a little bit more detail, I will slowly walk you through it. So we start, of course, with our MS analysis, where we then subsequently identify proteins, and we predominantly use MaxQuant as a way to identify proteins based on our spectra, and this allows us to capture a number of phosphosites. And we then try to focus on those that are quantifiable, And as you can see here, regardless of the organ, we predominantly capture serine phosphorylation, as is illustrated here in this pie chart, and we have also a portion of threonine and tyrosine phosphorylation. Now, when I say quantifiable proteins, this is a rather important concept, because when we want to apply statistics to this, we obviously need in both our conditions the same phosphopeptide detected in a number of independent biological replicates. So here we have a number of independent biological replicates. First example gives you the same phosphopeptide detected in all our control and all our treatment samples. There are a number of situations where we sometimes do not detect it in one biological replicate, but if we have sufficient values, we can still subject these to statistical analysis and look for phosphorylation events where there is a small up or down regulation with respect to that phosphorylation. On the other hand, there are examples where we do not detect one particular phosphopeptide in one treatment and then do detect it in the other treatment, which is really a black and white situation, which limits, of course, statistical analysis because we don't have values in one of these conditions, but on the other hand also reflects a very important change, because this particular phosphopeptide only appears in one condition. So this is very, very likely a very important regulatory component very much associated with the condition that we are looking at. So going back to this workflow, we then do find in our data set a number of phosphopeptides that are really only present in either the control or the treatment condition, which we then also include in our subsequent analysis. And then, of course, we have a number of values where we can do statistical analysis on and can really look at the phosphorylation profile in our treatment. So then we have a pool of unique proteins and a pool of significant proteins based on statistics that we can then use for further detailed analysis. And if we look at our data, we observe overlap with what we can identify between different organs, but we also see that there is a clear distinction between these different organs. And this is also with respect to the significantly up and down regulated ones, where we see that there are a number of common ones, and I'll come back to this a little bit later as well when we expand a bit more our data set. But there is also a number of likely or possibly organ-specific regulators, specific phosphorylation events that are going up and down in these wheat plants. So to dig a little bit deeper into this early temperature responsive wheat phosphoproteome, I want to give you a third example. And with this third example, we actually extended our data set by adding, in addition to these spikelets, we also added developing young anthers, with the idea that if we have these different organs that are all subjected to the same 1 hour treatment of high temperature, this would allow us to identify the core temperature signaling components and maybe also get further insight into temperature and organ specific responses. And indeed, as you can see in this Venn diagram, there are a number of proteins, phosphorylated proteins, phosphopeptides, that are common between all of the organs, but this is a rather low number. And these indeed reflect general temperature responses, for example, heat shock proteins, which are responding in all of these organs. There are, of course, also a large number of organ-specific ones, which is kind of indicating that if we want to get a good overview, a good handle on temperature signaling, we probably need to look in all of these organs independently and then try to map the signaling cascades that are associated with this response. Now, the way we approach this, and this is an example based on some of our recent data, is that we take, for example, seedlings, we expose them to high temperature, we subject them to this phosphoproteomics workflow, we then look for differential phosphorylation, and then, as I said, we use this difference in phosphorylation as a proxy as an indication for involvement in high temperature signaling. And then based on the profile in our data set, we identify a number of key candidates that we want to explore further, which we refer to as targets of temperature or TOT proteins. And I'll give you an example for one of these TOT proteins that we identified. So this one, which we called TOT3, the phosphostatus of this one is regulated at high temperature, so it is less phosphorylated at high temperature, and the phosphopeptide that we identified belongs to an uncharacterized kinase domain. So with this information, we assumed that it is likely that this kinase plays a role in temperature-mediated growth. So we therefore gained access to tilling population at the John Innes Center via Christopher Owe, and we could identify a number of lines which have an early stop codon, which we then started to characterize in a little bit more detail with respect to the response to high temperature. So if we look at these wheat seedlings, we observe that at the optimal, more or less optimal control temperature, 24 degrees, they are growing rather comparably. However, if we grow them at high temperature, and this holds true for both of the tilling lines that we looked at, the mutant line is much more responsive to the high temperature than our control line, which is nicely indicating that indeed, based on our phosphoproteome analysis, we can identify a number of candidates and then confirm, validate this with respect to the actual response in warm temperature mediated growth in this case. Now, of course, to bypass a little bit the limitations of wheat with respect to the speed of understanding the molecular responses and biochemical responses that are actually happening, we are then moving, as I mentioned earlier, for some of our mode of action studies to Arabidopsis. And it was very nice to see that if we identify loss of function alleles in Arabidopsis, and in this case we have two independent alleles, we see a rather similar picture as we saw in wheat. We have no obvious difference on the level of the young seedling at optimal temperature, in this case 21 degrees. But if we look then at the temperature that really controls thermomorphogenesis in Arabidopsis, and this is specifically elongation of the hypocotyl, the lengthening and angling of the petioles and leaves, we do see that while our control nicely responds to this, our mutant is less responsive to this treatment. And we can also very nicely quantify this, we can complement the mutant, and so on. So we can now use Arabidopsis to gain a little bit more understanding of this uncharacterized kinase by looking at its interacting partners, by looking at the downstream components, by, for example, doing then a new phosphoproteomics analysis on this mutant, which then really allows us to gain very detailed mechanistic understanding of this kinase, which we can then exploit again in wheat to see to what extent this protein there plays an important role. And with the fourth and final example, I want to also illustrate that similarly to what I've shown you with these wheat varieties in the beginning of my presentation, that we can also capture marker proteins for temperature tolerance and sensitivity. So, as I've already shown you, there is a response to high temperature, which is either growth promoting, the development and the growth of the young seedling leaves is affected. On the other hand, if the temperature becomes too high, there is a growth repressive effect, and we start to see yellowing leaves. So what we now reasoned is that if we would look at a more tolerant versus a more sensitive variety, we should be able to, on the one hand, capture important regulatory proteins for warm temperature mediated growth and responses. And on the other hand, try to identify markers within these sensitive and tolerant varieties that reflect the behavior of these plants, mature plants, to an environmental change, an increase in temperature in this case, which we could then, of course, when we know which of these markers plays an important role to kind of distinguish these varieties, these markers can then obviously also be used in targeted breeding approaches. So we looked at this sensitive and tolerant variety in a little bit more detail, and this is in the context of leaf length here, where we observe that indeed for the tolerant variety we get this kind of nice increase in growth until a certain temperature, and when the temperature becomes too high, growth is arrested, which we obviously also see in the sensitive variety. But we see that the sensitive variety, in contrast to the growth promotion that we get in the tolerant one, is not doing very well already under more temperatures that are growth promoting in the other variety. So we see that this is becoming much more earlier growth repressive compared to the tolerant variety. So we then subjected these two varieties essentially to our phosphoproteome analysis, and while we could identify a number of overlapping proteins, overlapping phosphopeptides, phosphorylated proteins in these two varieties, we also have a number that seem to be distinct between the two. What was actually interesting with respect to the overlapping ones, that we could identify a couple of proteins that were clearly showing opposite behavior. While they were either downregulated in the tolerant variety, these were then upregulated in the more sensitive variety, or vice versa, which is indicating that these proteins might explain the different response of these varieties to this specific temperature treatment. Again, suggesting that these might be ideal breeding targets to turn this sensitive variety into a more tolerant variety by, for example, knocking out these proteins, or more specifically modifying the phosphorylation status of these proteins in these different varieties. So in conclusion, what I have shown you today during this webinar is that we can use the wheat phosphoproteome to, on the one hand, identify signaling components, and in the context of TOT3 I have illustrated this with the temperature example, but we can also use the wheat phosphoproteome to identify novel breeding markers. So if we compare tolerant versus sensitive varieties, we can actually look at contrasting responses and then try to estimate which ones of these we need to introduce into the sensitive variety to make it tolerant, or vice versa. And one of the next steps that we are taking, because all the examples I have shown you today are associated with plant material that has been grown in glass houses or in growth chambers, we are now trying to move this actually to the field. And then I want to end with this quote from Eisenhower, who said that farming looks mighty easy when your plow is a pencil and you're 1,000 miles from the cornfield. And I kind of took the liberty to replace that with wheat here. So we really need, as a next step, to move all of these analysis into the field and to try and capture the dynamics of phosphorylation in a kind of more general context, but also upon very specific natural perturbations, including an increase in temperature. Which basically leaves me to end the presentation with thanking the people involved, which for the wheat work are mainly Casio, Tingting, and Pia, and there was some work done before in the context of wheat, but mainly in the context of our phosphoproteomics workflow by Elizabeth and Dai. And there are, of course, a number of collaborators, and the list is longer than the ones I have put up here, that are intimately involved in this project. So if you want to know more about what we are doing with respect to phosphorylation, with respect to phosphoproteomics, you can take a look at our website, send me an email, follow us on Twitter or Facebook, and hopefully I get to hear or see one of you again at some point. So I want to end the presentation with taking some questions, if there are any. I don't see any questions in the webinar control panel, so maybe there are no detailed questions or maybe more general questions that people had.
So thank you very much, Yves. This is Kelly. There is one question. What kind of database or proteome library could be used to map the proteins after MS analysis in wheat?
So what we've used is the IWGSC version 1 proteome database that was generated. So that's basically the starting point. This is then in silico digested, and then we are assessing using this MaxQuant software to map the spectra to the in silico, to the kind of the really observed peptides, to the ones that are predicted in silico. Of course, our assignment very much relies on the quality of identifying the proteins in this genome database. So with further improvements to this, with more detailed information on which proteins can actually be predicted to exist, the identification will of course become more global.
Then I have one question. Yves, you mentioned about going, really the next step is to translate this work to the field.
Yeah.
What do you see as those next steps?
So what we're doing now, because if you're doing this in a very much controlled environment, you're really simplifying your system. Because if you look at a field, there are so many combined stresses that are acting on these plants that this all needs to be integrated. So what we are currently doing in the lab is only varying the temperature response, and all the rest is rather controlled and rather sterile. So we try not to have biotic stresses. We try not to have drought stress in together with this high temperature stress. So we really try to very specifically look for what is affected by. We also have the microbiome in the soil that will impact. We will have various biotic and abiotic stress conditions that will impact the growth behavior and the responses of these plants, which of course, if you then on top add a temperature stress, this all needs to be very much integrated. So we're now, instead of trying to do this under very much controlled conditions, we are really trying to grow plants in the field and then monitor the conditions as much as possible, and then impose a temperature stress in a more natural setting.
Great. I don't see any other questions at this time. So Eve, I'd like to thank you again for a great webinar and for kicking off the IWGSC webinar series. And I'd also like to thank all of those who have attended today and who will be looking at the video in the future. So thank you very much.
Okay, you're welcome. Thank you. Bye-bye.